Let's talk about an animal that breaks a lot of rules in biology, the marbled crayfish. This freshwater species doesn't need a mate to reproduce. It is made entirely of clones and might be the only animal we know of that became a new species instantly through a process called saltational speciation.
The marbled crayfish, or Procambarus virginalis, is classified under the kingdom Animalia and belongs to the phylum Arthropoda. It's part of the class Malacostraca, order Decapoda, and family Cambieridae, placing it within a group of freshwater crayfish primarily found in North America, Europe, and Madagascar. Its genus is Procambarus, and the species name virginalis highlights its unique ability to reproduce through parthenogenesis.
Crustaceans are defined by a few key characteristics. They have a hard exoskeleton, jointed appendages, and have a body divided into three distinct regions: the head, thorax, and abdomen. In many species, the head and thorax are fused together, forming something called a cephalothorax, which provides extra protection. Most crustaceans are aquatic, but a few have adapted to land. Of the 40,000 known aquatic species, about 10% live in freshwater, including crayfish. Marbled crayfish are an invasive freshwater crustacean known for their rapid reproduction and adaptability. They eat algae, plants, snails, and amphibians, often outcompeting native species. They grow to 4 to 5 inches long with a marbled shell, typically olive to dark brown, but sometimes tan or reddish blue. They thrive in streams, rivers, ditches, and ponds, but they can burrow into soil to survive droughts, making them highly adaptable. Marbled crayfish reproduce asexually through cloning and were first discovered in 1995. Their exact origins are unknown, but they likely mutated from the slow crayfish in a German aquarium. They are fast swimmers, aggressive predators, and reproduce rapidly, making them a highly invasive species that threatens native ecosystems. The marbled crayfish are an all-female species that reproduce asexually, creating clones of themselves. They originated from Procambarus phalanx, but differ reproductively due to having extra chromosome copies. Analysis confirms they have a single origin and are triploid, meaning they possess 3 sets of chromosomes from the same species, a result of something called autoploidity, a rare genetic duplication process. These differences give them evolutionary advantages that include larger size, higher reproduction rates, and unique gene expression, allowing them to spread rapidly. The study supports classifying marbled crayfish as an independent asexual species and highlights them as a rare model for studying asexual reproduction, cloning, and rapid species formation. Researchers used 6 key tests to study the marbled crayfish's origins and unique traits. They used crossbreeding experiments, mitochondrial genome analysis, flow cytometry, microsatellite analysis, DNA methylationases, and morphological and life history analysis. This study found 6 key results that define the marbled crayfish's unique biology. The first, reproduction. They can mate with P. phalanx, but no hybrid offspring are produced. Instead, they reproduce by cloning themselves. Second, genetic identity. All marbled crayfish have identical DNA, proving that they came from a single colonial ancestor. The third, triploidy. They have 1.4 times more DNA than P. phalanx, confirming that they are triploid, with 3 sets of chromosomes instead of 2. The fourth, epigenetics. They have lower DNA methylation, suggesting differences in gene regulation and adaptability. The fifth, traits and fitness. They grow faster, get bigger, live longer, and produce more offspring than P. phalanx, giving them a strong survival advantage. The sixth, evolutionary significance. They represent a rare, rapid speciation event, evolving through autoploidy and parthenogenesis.
In the discussion section of the study, the researchers concluded that the marbled crayfish meets all the criteria for being its own species. This is due to differences between marbled crayfish and their mother species, P. phalanx, specifically due to the differences in reproduction, gene expression, and the marbled crayfish having superior life history traits, like larger bodies, living longer, increased offspring production, and geographical isolation. The researchers also confirmed that they have a single origin, meaning all marbled crayfish organisms from aquariums to the wild originated from one singular mother organism, which is a unique evolutionary history. The researchers also theorized that marbled crayfish could represent a new model for cloning in animals. Marbled crayfish offer an interesting model for how a species can be formed so quickly by asexual reproduction, and how much genetic change needs to occur for a new species to form. The research done in the study is very significant for the possibility of future studies involving the marbled crayfish. As mentioned in the discussion section, the marbled crayfish is a unique anomaly in the animal kingdom and could be used as a model for studying rapid species formation, as well as the genetic changes that go along with species formation. This is the basis for evolutionary studies as we know it, and could contribute to new theories and ideas about the evolving animal world around us. On top of this, the marbled crayfish's unique reproduction method offers a model for the study of tumor parthenogenesis, or asexual cloning, and the many adaptations tumors make in the human body. This animal is a very rare example of asexual cloning and well-developed adaptations to different environments, which are common traits among cancerous tumors. Understanding these early stages of asexual cloning can possibly help us understand early tumor development and evolution, which is important for cancer treatments and the understanding of cancer development as a whole.
These are the sources we used, and thank you for listening.